So this is going to be work that I did in my postdoc, not the work that I've done in the last 6 months in my new lab, unfortunately. But yeah, so I wanted to start first by this image here, which is a graphical representation of how I think about microbes, and that's in these dense and diverse microbial communities called microbiomes. A lot of us are well accustomed to thinking about these, but they have many different they impact our lives in many different ways. And I'm really interested in the gut microbiome, as Jeremy sort of introduced already. And there's a lot of benefits of the microbiome to us. And so thinking about how the composition of the microbiome affects its function is very important, because it's very important for our health. But microbiomes are not necessarily only full of beneficial bacteria, and they can also be reservoirs of harmful strains, and that can include antimicrobial resistant strains and opportunistic pathogens. And so really what we want to think about is also, aside from harnessing the benefits of microbiomes, how we can rationally engineer its composition to specifically remove strains that can potentially cause us harm. And so this is the kind of research question we set out to address. And when you think about removing a strain, often you think about an antibiotic, and that can be very useful and in fact essential in some cases. But the problem is that antibiotics are often broad in spectrum. And so what that means is you can decimate your microbiome and you're not left with a lot of these beneficial functions that come with it. So what we want to try and achieve is a way to engineer a microbiome where we can specifically target one species without having to mess with the whole microbiome. So that does sound like a challenging problem, and I think it is, but we can get some clues by thinking about microbiomes and thinking about how microbes naturally live. And so we can think about what they compete over, for example, and what determines the abundance of certain species. And competition for resources is a critical factor. And in fact, we showed in recent work that the ability of a certain microbe to acquire a nutrient will be very deterministic in its ability to colonize a microbiome. But there's actually a whole nother form of competition called interference competition. And bacteria actually have an amazing arsenal of different weapons that they can use to antagonize each other. And they range in the kind of flavor. And I just want to show you two examples here. One is these diffusible toxins, and examples are colicins of E. coli. They've been really well studied for decades. But what you can see here is two colonies of E. coli side by side. And in the middle, you have this big zone of death. And that's actually the colicins antagonizing each other. So both of these colonies produce these toxins. And so they're incredibly potent. And you also have contact-dependent weapons, and that includes type 6 secretion systems, which many of you are probably familiar with, such as shown here. This is the type 6 of Acinetobacter bailii, which is very much antagonizing this strain of E. coli. So they're really potent, these weapons. And what's also very important about them is that they're often narrow spectrum. And that's because they're evolved to target competitors of niche competitors. So if you eat the same nutrients, you want to evolve a weapon to target them. And so it makes more sense to not necessarily spend energy targeting microbes that you're not going to be competing with. And so the idea here is, if we can try and leverage these for our own benefit, yeah, let's try to do that. But that's obviously a big challenge. And we're trying to think of effectively how we can introduce a microbiome, a microbe that we might engineer to encode a mechanism of competition into a community that's going to be really dense and really occupied already. And so how are we going to do this? And so in order to address this question, which has a lot of different moving parts that go into it, we started by building a simple mathematical model. And it's actually really quite simple. You have nutrients coming in that can represent the diet of a human, for example. And then you have some sort of clearance, and within you can embed a community of microbes, or even more simply, just two species competing over nutrients. And in order to make this this model sort of recapitulate the natural ecology of of the microbiome, we wanted to keep some key aspects. And one of them is this idea of strain invasions. And so when you think about what a strain would need to enter a microbiome and potentially displace another, you have this asymmetry. You have a rare strain that's going to come in and potentially lead to a turnover event. So we wanted to keep this asymmetry. And we wanted to represent the importance of competition for nutrients, because we had shown in that previous study that it was so important in determining composition. And so what we do here is we allow both these strains to compete over a shared nutrient, but also invest in their own private nutrient. And that's actually really representative of what happens in nature when you think about a strain like E. coli. A species like E. coli, you have a lot of different strains that vary in their metabolic capacity. And so we can put some differential equations to that and run some simple simulations. This is done with the help of Veit, who's a brilliant mathematician. And what you can see in this scenario, where you have a single strain that invades into a niche that's occupied by another species, but they use the same nutrient, you get something you'd probably expect where you have no invasion. But this changes dramatically the moment that you allow the metabolic differences to kind of partition the niche a little, and now they can use their own nutrient. And now we can ask, how does interference competition, that second form of competition, play into this? We can allow each of these invading strains to now produce a toxin and run the same simulations again. And what you can see on the left here is that effectively nothing's changed, but you see a very different picture on the right. Now all of a sudden the strain is very successful at invading because of its metabolic difference, but it now reaches a density to which it can produce a large amount of toxin and be very successful at displacing the other strain. And so that's really cool, because now we have this prediction just based on thinking about the natural ecology of these microbes and the forms of competition they use. And our prediction is as follows, which is that if you have differences in the metabolism of competing species, it should actually empower the use of toxins. And that's now quite interesting when we think of this first question of how we can rationally engineer a microbiome. And this starts to become something we maybe can use therapeutically. And so we wanted to test this prediction, but we wanted to add a little complexity, because I've been telling you over and over again that I'm interested in microbiomes and that strains compete within these diverse communities. And so we did an experiment, and this was done with the help of my former master student, Megan. And we basically set up this experiment where you would have a resident strain that you would try and target, and you'd embed that in a community. And we can grow all these strains in the lab, in these anaerobic chambers that Jeremy was highlighting. But you can culture them all, and you can mix them together. And then a few hours later, 24 hours, you can add a second strain. And we didn't want to just use lab strains. We wanted to target some species that we really cared about. And so we used antimicrobial resistant E. coli as targets. So we specifically these extended spectrum beta-lactamase that we got from a hospital in the UK. And we basically put those as the resident strains embedded within the community. And then as invading strains, we got another set of E. coli that we isolated from healthy humans, and then engineered them to produce a toxin. And so what we did what you saw in the experiment is, as we predicted in the model, the metabolic differences between the two strains allowed one to invade in the presence of the other, and then it displaced it once it's reached a certain density dependent on its toxin. But then when you add the community, all of a sudden this effect goes away. And to try and understand that, we turn back to our previous data where we realized that really what the community is doing is kind of occupying all these extra niches. So you imagine that these two strains, these two E. coli strains, would represent perhaps this pairwise interaction between this yellow species and this red pathogen coming in. But when you have the rest of the community, all these free niches that the pathogen can normally use, or the invading strain in this case, are kind of blocked. And so we could come up with this idea that perhaps really what we need to get a strain to effectively remove another one is we need to identify nutrients that are kind of unique relative to the entire community, such that the invading strain can grow and then displace another one. And of course, this is again a very testable prediction, because you can go into the lab and look at that community we had been working with, and look at that antimicrobial resistant E. coli, and we can profile the all the carbon sources that, or at least a large array of them, using these biolog plates, and we can basically look, what are those some nutrients that are invader specific, and then we can supplement that into the media, repeat the experiment, and challenge again with these E. coli engineered to express these toxins. And so now what I'm showing you on the left side here is just the density of the target strain, so this antimicrobial resistant E. coli. So in the absence of a nutrient that's private for the invading strain, the target species never gets affected, regardless of whether the invader has a toxin. But the situation is very different when you now combine the two forms of competition. You allow the strain to first grow based on a specific nutrient, and then you get the toxin to actually very effectively displace the other strain. And so altogether, this kind of shows how important thinking about microbial competition in ecology is for trying to engineer microbiomes. You have all these complex ecological networks that can be kind of simplified if you look at the pieces that kind of define the rules of success in microbiomes. And what I've shown here is that when you want to, engraft a strain or get a strain into a community, you really need to think about nutrient capabilities and overlap. And if you were to then try and displace another one, you can leverage other forms of competition like interference competition. And with that, I want to thank everyone in my former lab who has been very instrumental in this project. And now in my new lab, I have one graduate student who has been brave enough to start the same day I did. And so hopefully we've got some exciting things coming out. But yeah, I'm happy to talk further in the Q&A session.